and I'll post it for you. So these are questions from the cell reading project, right? We all agree that that's what's going on?
Yes.
All right, so give me a question. Yeah.
Okay, so on page 73, it talks about the osmoregulation.
Osmoregulation, yeah, what about it?
I don't know what that means.
What that means. All right, so it's a good question. I like that some people said that. Okay, so people don't know what that means. Here's my suggestion in the future. What page was that?
73.
73. So my suggestion, anytime you come across a word you don't know, honestly, in your generation, it seems really easy. Google it, right? And it really quickly, you get answers. But let me give you the answer, because you said you were working on it last night, right? And so if I was working on it last night and I was at home, and assuming I have computer access, and I was working on the question, I would just quickly Google, what is osmoregulation? You could get an answer, but it would take a little bit of work, wouldn't it? And probably, I'd say about half an hour to an hour to really figure out what the heck it is, but I can do it much faster for you. So the word is osmoregulation. Now, we haven't talked about this yet. The regulation, what do you think regulation means? Keeping it the same. That's good, right? We talked about that, right? That's one of the qualities of life, is regulating different qualities of life, right? Temperature, we called it homeostasis, right? Keeping homeostasis, keeping everything the same. Osmome is water concentration, right? So it has to do with water, movement of water, and of course regulation, keeping it the same, all right? Keeping the same. So that's what we're talking about. And now there's osmoregulation in many different, at many different levels. You, for instance, have something called a bladder. You have two kidneys. Everybody knows what those kidneys are, right? Horrible drawings, I know. And they both feed in through various tubes into something called a bladder. And now the bladder then feeds in through a urethra, and of course that's where you go to the bathroom and you kind of urinate out into the toilet, right? Now that doesn't sound like, it sounds kind of like ew, gross, but it's not gross. It's actually very important. And once your kidneys go, you have a very short time to live, unless we can give you something called dialysis, which will help with that. But dialysis, unfortunately, can only be done for a while before you start to run out of time. You need to have a kidney transplant. So what the kidneys do is they filter your blood. They get all the waste from your body out of your blood, and they put it into the urine. That's what we call urethra. Urine. Urine is actually what would be ammonia if our bodies didn't change the ammonia into urea. Urea is kind of is our nitrogenous waste. All the nitrogen, all the protein and nucleic acid, all the nitrogen we produce, the waste has to be put out of our body. Otherwise, it's poison to us. That's why we use ammonia to sanitize surfaces, don't we? You can use ammonia to kill bacteria and kill just about anything, because ammonia is a poison. And so we need to get rid of this substance called urea, because urea is a poison. We sweat urea out. That's one way we get rid of it. But the big way is we put it into a bladder. Now, again, these drawings are horrible because a kidney does not look like that. It looks more like something like that, right? Right, so it has more of a kidney bean shape. That's why we call it kidney bean, a kidney bean. So they filters out, and the bladder looks a little wrong too, but that's okay. These are tubes, right? And so we regulate our water, right, as an organism. So as a big organism, we regulate our water. All mammals do, right? There's all kinds of ways organisms regulate their water. Now, that's how we do it, and so that obviously we lose water, right, through when we urinate and sweat, and so one thing we do is we drink, and by drinking water, we replace the water we lost. That's how we regulate. That's how humans do it. Now, think about a cell. a cell, and pick a cell. Somebody give me, I have it here. What page was that on again? 73. Now let's take a look at 73. If anyone who wants to kind of figure this out with us, go ahead and take a look at 73 for a second. Tell me the kinds of cells that they're talking about when they're talking about osmoregulating. They're unicellular, that's right. They're all this. Well, if it's a cell, these organisms are unicellular, that's right. And this one is called, what's this one called? Do you recognize what this one is? Am I drawing it okay? The euglena, that's right. And the euglena has this, I'm going to do a different color set. So I'm going to say, okay, let's make this red. Let's call red the nucleus. And so this thing is called euglena. Who can tell me something about euglena as a single-celled organism? First of all, quickly, is it prokaryote or is it eukaryote? Eukaryote, that's right. Somebody, you noticed it right away, it's a nucleus, so we're good. The next question, and of course it has an eye spot. Who can tell me what an eye spot is? What do you think an eye spot is? Obviously a different color.
It detects light.
It detects light, that's right. That's why we call it an eye spot. It's not an eye, because an eye is an organ, right? If you think about an eye, it's round, it has a nerve, it's actually, an eye is an extension of your brain, has a retina, has an iris, right? A pupil, pigment. So it's made of many, many cells. This is only one cell, so it can't be an eye. But it does detect light. And it's very simple. It doesn't really, as far as I know, it doesn't really detect shapes. It just says, is it dark or is it light? That's all. So that's why we call it an eye spot. Now it has this thing in the center, this organelle. It's a common organelle, especially in plants, and you know it is a central, in many different single-celled organisms. We have this thing called a vacuole. In plants, it's called the central vacuole. Plant cells are gigantic, and when you get to plant cells in the packet, you'll see how big it is. But there's this organelle that acts a lot like a bladder, in that it swells up and then it urinates out, in a way, water. It doesn't really urinate because it's called the contractile vacuole. Who knows what the word vacuole means? Who looked it up? The word vacuole. It is in plant cells. I said, yeah, that's right. Vacuoles, central vacuoles are in plant cells. That's good. Yes, I mentioned that a minute ago, but you're absolutely right. And again, I don't want to use names right now because we're recording, so let's not use names. But yes, the vacuole isn't. We do have vacuoles in plant cells. Now think about it. Why does that make sense? Why do you have a vacuole that helps regulate water in single-celled organisms, and you have a vacuole that helps regulate water in plants, but not in animals?
So, regulations like
Balance the water. Why does a plant need to have something in every one of its cells to be able to take care of extra water or deal with the lack of water? What can an animal do that a plant can't? Go get water. Excellent. Perfect. That's right. A plant can't go get water. So if it's dry the way it's been over the last few days, how does that plant continue to work if it doesn't get water? It has water where? In the vacuole. That's right. Some of you are getting it, right? So the vacuole ends up being how plants and single-celled organisms store water and deal with excess water. So this vacuole, by the way, the vacuole, you've heard of the word vacuum? What does vacuum mean? Okay, a vacuum sucks up, but actually it doesn't suck up. That's interesting. There's an old saying, science does not suck. Because there's no such thing as suction. Did you know that?
Oh.
There's no such thing as suction. Something interesting out from physics. You're not sucking. What's happening is you created a vacuum. So let me be clear on this. Let me pause this for a second. So in a vacuum just means empty space. You should write that down because I'm sure that you're going to see it in physics. I'm sure you're going to see it on the ACT. It could be on the ACT somewhere. A vacuum means empty space. And what happens in nature, and there's another old saying that nature abhors an empty space, abhors a vacuum. And what that means is that in nature, if there's empty space, nature fills it up. So it's almost a force, and we're going to talk about that, how things move from high to low. So vacuums are not empty for very long. And it turns out we call these vacuoles because they're small empty spaces, right? Now you might ask, well, how is it that I can suck up water through a straw, right? Isn't that what someone said earlier? How is it when I suck up on a straw, I'm not sucking? Well, that's true, you're not. What it is you have a glass and you have some liquid in it, and you have a straw it's an empty space. That's not empty, actually, it's full of air. And you put your mouth to this end, right? This is where you're putting your mouth, on this end. And then you empty your lungs. Your lungs are empty. Now, what is pushing down on everything all the time? Air pressure. So here's air pressure pushing down in every direction on this water. Now you have an empty space over here on this side, empty. What does nature do to empty spaces? It fills them up. And so the air pressure is pushing on the water constantly. You empty this space here, right? This is all gone because you emptied your lungs. So now it's all gone. So what the air pressure does is it pushes the water up and into your mouth. And before it gets into your lungs, you close the little flap in the back of your throat, and the water goes down your throat, and then you take a breath. That's why you go, and when you're sucking, you're actually just emptying your lungs, and the air pressure pushes it up. That vacuum cleaner that you have at home, the only thing it's doing, that motor is just emptying the space inside the vacuum cleaner. And then at the other end of the vacuum cleaner, the opening that has where the brush is, all the air rushes in, and taking with it all the dust and garbage that the vacuum cleaner is picking up with it. And then you have a filter that keeps the garbage in the chamber, and the air gets pushed out the other side, creating more vacuum, pulling more air in. But there's no real pulling, it's always pushing. That's why we don't say suck in science. Yeah.
So why is it that when you drink from the straw, and then as the water is still in the straw, you hold the tip where you're drinking from the water doesn't pour out?
Great question. What is it that keeps the water in? Now think about it, if you can pinch off the straw, hold on a second. So the question was, why is it that you could take a straw, and I don't have a lot of time for this, so this was not the intent.
Answer the actual question.
Wow. So when you take the straw and you pinch this off here, and you have some liquid inside, right? That's the question. It's a real good question. So you have some liquid in there, right? How come the liquid doesn't fall back out? Well, what's happening on this end? What's pushing up, keeping it in there? Air pressure, right? It's what's pushing on all of us all the time. Air pressure is pushing up. Why is air pressure not pushing it down the other way? Because you pinched, it's being blocked. You pinched it off. So now air pressure is not allowing, there's no air pressure on this side, but there's air pressure pushing it up. So, and also you have something else called surface tension, right? You have cohesion going on, right? Water molecules are pulling on each other, so they're holding each other there. So a combination of cohesion and air pressure are keeping that liquid in that straw. All right? All right, so there you got, that explains that. Now, the actual question was, what is how do we osmoregulate? What is osmoregulation? And what some people would call a smart aleck question, but I don't think it was smart aleck. Did you actually answer the question? And I kind of did already, but I'll be clearer by spelling it out for you. And that's that water, how do single-celled organisms regulate their water? How do they osmoregulate, right? And the way they osmoregulate, in the case of euglena, is what? Who's paid attention and knows the answer? Yeah. Does someone, when the next person comes back, how do they do it?
By using the contractile vacuole.
Exactly, by using the contractile vacuole. So actually I did answer the question, and that is that using the contractile vacuole allows the euglena to regulate water. So when there's a lot of water, when there's a lot of water, let's go ahead and make that chart. A lot of water. What the contractile vacuole does what? But when there's too much water, it's a single-celled organism, there's too much water, oh my God, what am I gonna do? I'm gonna drown. What's the cell gonna do? What's that empty space called the contractile vacuole, what's it gonna do? It's gonna what? Push the water out? That's a good idea, but it's inside the cell. It's going to swell up. So when that, oh man, I can't believe it did that. So when you're talking about a contractile vacuole, you're talking about when there's a lot of water, the contractile vacuole swells up. It's really big. That's because it's taking all the extra water into itself. So it regulates the concentration of water by pulling water out of the cell. And where's all the water going into? What's going on? Well, the water's rushing in here, in this space, right, in the cytoplasm. The contractile vacuole sucks it all up, right? In other words, it allows the water to come in and traps it in itself. So now when there's not a lot of water, not a lot, what does the contractile vacuole do? Contractile vacuole. When there's not a lot of water, when the cell starts to lose its concentration of water in here, what's this contractile vacuole going to do? Anybody? No, nothing. Think about it. It took it in when there was a lot. What's it going to do when? Push it out. Nah, push it out. And that's exactly what it does. It doesn't really push, but in any case, it lets the water out. You see. And so when this is how it helps regulate the cell's water concentration. When there's a lot of water in this space, the contractile vacuole swells up. When there's not a lot of water in this space, it empties out. Now I spent time explaining this in this class. Guess what one of the questions on the test is going to be? About this vacuole and how it works. I will be ready to answer a question about that next week. Now that's great for euglena. What about amoeba? How do they do it? Contractile vacuole. How do paramecium do it? Contractile vacuole. How does the last one there do it? The chlamydomas. They're contractile vacuole. So no matter what, single-celled organisms generally regulate water, osmoregulate using contractile vacuoles in this process of where the vacuole swells up or empties out. That's the answer for osmoregulation for all of those. All right, that answered your question, yes? Okay. Now, what else is there? What other questions? I spent some time answering stuff that wasn't in the packet because you guys didn't, again, so let's go ahead and take a look at what this means. You have this cell, and you have this thing that looks like this. It's a big old... See, you guys are going to mess up on your test. I hope that your parents watch this video and they listen to this, and when they ask me, how come my kid doesn't understand, I hope they understand this because you're not listening. I really do. But it doesn't... I'm just going to go on. I'm going to continue. So here you have this stuff. You can't really tell in the picture, right? I'm just kind of sketching it. Just kind of haze, right? But you have this thing here that's labeled A. So this is a basic structure, they say. What is A? What's the big, dark, circular thing in the middle of a cell? Oh my gosh, yes, my goodness, it's the nucleus. That should be easy. And at this point, the third day working on this should be clear. Then they go ahead and they draw some more cells. So that's one cell. But then you have this other cell, and obviously, and there's kind of these cells. It's a plant. Everybody knows it's a plant cell because you have a giant plant cell drawing right above it. right? So then it has another one of these big dots, big circles in here, and then it has this thing labeled here, B. What's the thing that separates all these plant cells from each other? Cell wall. There's these big, thick things called cell walls. And that should be clear. Then there's these things called C. They're not dark. You have to look real closely at it.
It's the nucleolus.
No. No, I don't know what you're speaking.
It's the goal.
So there's this thing. It looks like an empty bag. An empty bag looks empty, clear. It's clear, right? It's big. It's not quite in the center, but it's big. It's clear. It's not dark like the nucleus. It's big and clear. No, the nucleolus is dark if you look at the plant. No, you can't see the Golgi. It's too small to see.
It's the starch.
Oh, it's starch.
Starch granules would be small, actually, small and dark.
Oh, I thought that was good.
See how in the drawing they're dark?
Yeah. Is it the central?
It's a large central vacuole. That's right.
It's the big thing in the middle.
Where is it?
It's C. That's C.
It's the big thing in the middle.
So C's in there. It's big. Is it as big as in the drawing there? No, but it's big. It's clear. Now D is there's a lot of them. Chloroplast. There you go. In a plant cell, there's going to be a lot of these, and they look oblong. They look a lot like mitochondria from under a microscope like this, in that they're oblong, almost like little beans. And there's a lot of them because you need a lot of them to do photosynthesis, and we're called these organelles chloroplasts. All right. And that's D.
Like three of them, like how they set up in the.
No, they just pointed to three of them. You look at the cell, you'll see there's a bunch of them. I want to say there's something like 40 in the picture.
No, I mean like three, you know,
The what?
I can't explain it.
Let's do the animal.
Like, is it supposed to be only three?
Three what?
Three of those, like points in the chloroplast?
I'm not sure what you guys are talking about.
Three of these.
No, there's not always three. Those are called thylakoid membranes. There's more than three. They just draw it that way. All right. Well, that's it. Time's up for class. How many millimeters in a meter? 1,000. I got it right off that chart. Did you have to memorize it? No. You should. You will have to in the future. But in this class on the test, I'm going to give you. You're welcome. I'm going to give you the equivalencies. I will probably give you the conversion factor. Okay? So I will give you the equivalencies, and then just like I will on the practice test, I'll give you the equivalency, and then I'll tell you to convert it. Okay? I could give you anything as long as I tell you what's equal. You'll see how easy it is once I show you. Okay? So 1 meter is 1,000 millimeters, or 1,000 millimeters is equal to a meter. How many micrometers in a meter? 1 million. 1 million. How many nanometers in a meter? 1 billion, right? Now, let's go off the chart. What do we want to do? Are we going to convert to meters? Are we converting to meters at all? No, we're not. We're not converting to meters. We're converting from micrometers, which is what the scale the pictures are in, and we're going to convert to what? To millimeters. So the question is, how many micrometers in a millimeter? How many micrometers in a millimeter? Anybody? 1,000. So there's 1,000 micrometers in a millimeter. Now comes the listening carefully part. The people that listen carefully This will be as easy as it can get. This is an equal sign. That means this is the same as this. Do you agree with me? If you don't see that, you need to say something now. Because if you will not understand everything else after this. When you see this, and then you see an equal sign, you see this, that means every time I have 1,000 micrometers, it's exactly the same as 1 millimeter. Agreed? Okay. So, because they're the same, do you agree that I could do this? 1,000 micrometers over 1 millimeter, and that equals 1? Right? If I put 10 over 10, what is it? If I put A over A, what is it? So if I put 1,000 micrometer over 1 millimeter, what is it? Why is it 1? Because they're the same. Only because these two are equal, right? You need your equivalencies. That's why on the chart in the workbook it says equivalency, right? Equivalent. Are we all good up to now? Yeah. I'm not going. I think this is pretty straightforward. Now, look at the rules I told you to write down off the board, right? Number 1, find your equivalency. Find your conversion factor. This is your conversion factor. This is what you're going to use to convert. You're going to do that by multiplying. We're going to multiply. We're going to take this thing, and I'm going to use parentheses. You're going to take this conversion factor. How did we find the conversion factor? We asked, okay, I got micrometers and I need to go to millimeters. How am I going to do that? I need something there. I need an equivalence. I need to know something's equal to something. Once I know they're equal, I can put one over the other, and I can use that to multiply. And that's going to help me change my units in a problem. That's my factor, my conversion factor. Once you have this, you're ready to go. Is everybody good up to now? All right, so if you're confused about something, it's going to be a time to ask. Now would be good. All right. I don't know why this is all broken up, but it is. All right, so now let's go ahead and do the problem, because that's all we need to know to do the problem. Pick a problem. Let me know which one you want to do. Number 2. Or you mean 1, I'm guessing you're saying 1B. All right, so who measured 1B? Who has a number for 1B? Nobody measured 1B? So I guess we can't do 1B. I guess I'll just make up a number. You guys have to measure it. You guys have to tell me. What is it? For B, 1B is the units are, the unit that they have, the little line they have is 100 micrometers. So it has to be 200 or 250 micrometers is what you're saying to me? Is that what you're saying? I don't know what you're saying. All right, so you're saying 200 micrometers? Is that what everybody agrees? Does someone measure it? So you think about 200 micrometers? Okay, let's go ahead and use that. You think it's 300 micrometers? Okay, so let's go 300 micrometers, 300. That's what we have. What did the rules that I put on the board, what did it say to do? To write it on the which side, the left or right? Right, left. So this is left. In case you don't know, this is left. Some people don't know, so I'm not laughing. I'm not being sarcastic. It's a thing. Some people confuse their left and the right. This is the left. I'm going to put an equal sign. And what's this side if that's left? That's the right side, right? What am I putting on the right side? What you want, the unit you want. What's the unit I want? Millimeters. Some number of millimeters. I don't know. I don't know how many 300 micrometers are. So you all understand, and do you agree that I can take this and put a 1 under it? Because I could do that with anything, right? So I'm just going to make everything into a fraction, because believe it or not, fractions are the easiest way to convert things, in my opinion. And this is a very common thing to do in physics and biology, chemistry. It's almost every science. You're going to do this the whole process is called dimensional analysis. Dimensional analysis. So there's your 300 micrometers. How am I going to get to millimeters? I told you I have to multiply. So by a factor, I know I have to have a division sign. Do you also agree, just one more point before I go ahead and explain this. Do you agree that 1,000 micrometers over 1 millimeter is the same as 1? Everybody agrees with that. And do you all agree that 1 millimeter over 1,000 micrometers is the same thing? Right? 10 over 10 is the same as 10 over 10, right? So since these are equal, I can put this on top or this on top. Doesn't matter because they're the same, right? Okay, as long as we're in agreement. Why? Because of this equivalency. So which one am I going to put on the bottom in order to cancel? Micrometer. It has to go on the bottom so it cancels. And so what's going to go on the top? Millimeter. How many micrometers in a millimeter? 1,000. And 1 goes on top. That's exactly right. And that's it. You're ready to go. Plug and chug. See how easy that was? Real easy. So now let's go ahead and do it. You take 300, you divide it by 1,000. I hope you can, if you can do it in your head, that's wonderful because it's really easy, right? Move the decimal 3 times. Or you could cross out the zeros. But if you cross out the zeros, 3 divided by 10, you still have to move the decimal. So it's 0.3 is the answer, 0.3 millimeters. See how easy that was? Question.
I feel like you could just, you see how many zeros in the micrometer, how many ever it is, you can just move, turn to zero, move over.
Yeah, that's what I would do too. But the reason I'm telling you to set this up is for a couple reasons. Number one, in this class, I'm not going to ask you to do a lot of dimensional analysis. But when you get to chemistry and physics, you're going to have to convert the number 5 or 6 times to get the unit you want. You have to have 5 or 6 factors in a row to get the unit you want. 5 or 6 cancellations, a bunch of multiplications and divisions. So learn the process so that you're ready for the next step in your education. But here's the other reason to do it is when you cancel, guess what you're doing? You're checking to make sure you're doing it right. Because you know what kids will do on a test? Instead of dividing by 1,000, guess what they'll do? They'll multiply by 1,000.
That's what I was about to do at first.
Yeah, that's what's going to happen. And what this process does is by analyzing the dimensions, you're able to see, okay, I know I'm right because, see, I got millimeters up here and I got millimeters at the end. If I had it reversed, I would have micrometers. I wouldn't be able to cancel, and I'd have some crazy answer. And I know therefore I'm doing something wrong. Does that make sense? All right. Now, do you want to do a couple more problems, practice, just to make sure we got it? Do you want to? Okay, because I want to. Yeah. I'm going to make the others up. I'm going to make the others up so that. If you got it and you're ready to go ahead. And I'm going to go over some more answers, some more things that are a little confusing. I'm going to show you a small video as well. All right. A short video on centrifugation, that spinning thing that you have to figure out. So let's go ahead and do this. Let's say I give you 3 micrometers. And I want to know how many let me see, nanometers is that? All right, how many nanometers is that? So let's do the equivalency. What's the equivalency? First thing I got to do is find the equivalency and therefore the conversion factor. And the equivalency is 1 micrometer equals 1,000 nanometers, right? One micrometer, remember that weird U, is equal to 1,000 millimeters. Everybody agreed with that? Is that what that says? So that means my conversion factor can be one or the other. It can be 1 micrometer over 1,000 Millimeters, oh, I'm putting millimeters, I'm sorry. This shouldn't be millimeters, this should be nanometers. Nobody caught that, how come nobody caught it?
I did that.
You didn't say anything, okay. Next time, say something, it saves us all time. So 1 micrometer equals 1,000 nanometers, so I have two possible conversion factors from that. 1 micrometer over 1,000 nanometers, or 1,000 nanometers over 1 micrometer. So those are my two possible conversion factors I can use. Which ones am I going to use? Well, I don't know. It depends what I want and what I have. I have 3 micrometers, and I need millimeters. How do I get there? I need a conversion factor. Which is going to go on the bottom? Well, first of all, let me remember to put this over 1 so it's more obvious to me what's going on. What do I need to put at the bottom? I need micrometer at the bottom because that's what's going to cancel, and I have nanometers on top. So micrometers cancel. I'm good with that, so that works for me. So how many micrometers in a nanometer? It's 1,000. 1 micrometer. There's 1,000 nanometers. Now I'm multiplying, aren't I? Just like that. Boom. I just changed the problem. So I could easily make 2 versions of the test where one I ask you to multiply it times that number, and the other divide by that, just by simply asking you to go one way or the other, keeping all the numbers the same. So I want, and I put millimeters here again. Nobody caught that. Nobody said anything. Dang it, guys. Help me out here. It's nanometers. So what's the answer? Should be easy. Everybody should be able to do this one. 3,000. Boom. Is that hard? So this is really easy, right? As long as you set it up correctly, it's easy. Do you agree? All right. Do you need me to do another one, or are we good? All right. So if you want help while we're doing it, you can come up here and ask some questions. I just recorded it so I could send it off to you this evening as well. All right. So let's do some answers down here. Let's go through, and I'm going to go ahead and not... I guess I might as well stop recording, so this is going to be... Because I thought it was a really good question. I thought people could get it, but I think there's a good confusion about what autotrophs are and what heterotrophs are. And I'm looking at page 73. And autotrophs mean they're organisms that make their own food. So there's several ways that you can do that, and then there's heterotrophs. They eat food. They eat others' food from others. So autotrophs, most autotrophs on this planet, right? They use something called photosynthesis. There are chemotrophs that use chemicals, and they can get through chemical reactions. They can make their own energy. But autotrophs on this planet, what is it that they need in order to make their own food? Say again? Sunlight. And a couple of you had that answer. Sunlight. They need light. Autotrophs, and this is key, need light. Most autotrophs need light. So the question is, suggest why an autotroph would need an eye spot. What does an eye spot do? Exactly, to detect light. Because it's going to want to be where there's light so that it can make its own food. So that's pretty straightforward, isn't it? Also remember, as you're doing these, the answers are right in front of you. In a lot of this, the answers are right in front of you. If we look at fungal cells, what I'd like you to do, if you really want to understand the difference between yeast and single-cell fungi and filamentous fungi, make a Venn diagram. Make a Venn diagram. I had a couple students make it this morning, where you take 2 circles, and you take single cell. fungi. And what's an example of a single-celled fungi? Yeast, right? And over here on this other circle is going to be all the characteristics of a filamentous. What does it mean, filamentous? What do you think it means, filamentous? Anybody? Nobody has a guess? Look at the word. What does it sound like? Have you ever heard of the word filament?
No.
You've never heard of the word filament? You better write it down. You better learn it. Filament is a string, a thin string. Molds can be very much like thin strings. You ever see a mold growing on bread? It looks fuzzy, hairy, right? That's because they're filamentous fungi. Yeast don't do that. Yeast don't make filaments. So in the middle, put what's common between the two. And on the other two, put what's different between the two. Oh, I put 3 F's in different. Is that nice? That is different, actually. The common characteristics in the middle, the different characteristics on each side. That'll help you understand and answer these questions. All right? So that's for fungi. Also, please notice that fungi are eukaryotic. So that's one thing you can put in the common. They're all eukaryotic. They all have nuclei. They have a nucleus. But they're also not, they're all heterotrophs. They all eat. They usually eat dead things. That's what fungi usually do. They're saccharivores, right? They help get rid of dead organic materials. Okay, so the next page, page 75, we already went through the answers of what the actual pictures were. You looked at plant cells, you looked at the major structures. They're there. You need to use those for the next few pages, and the same with the animal cells. There's a question. Yes, I don't want to say your name because we're recording.
On page 75, I don't know, what's, cytoplasmic stream?
Right, where is that? Where do you see that?
2.
Number 2. Okay, very interesting. So the cytoplasm can move, and we call that streaming. In other words, like a stream, like a movement down from one end of the cytoplasm to the other. What you can see here, you can't see it here in the picture, but if we had a movie, you could see cytoplasmic streaming. And so cytoplasmic streaming literally is when you have the cytoplasm moving, and what you'll see is the chloroplasts will be moving from one end of the cell to the other. Any other questions? All right, so let's go ahead, and the next animal cells, pretty straightforward. Nobody has an issue with those. We went through. I want to emphasize again, please look at B on page 76. Those are 2 photographs. You see how hard the photographs are to be able to identify, right? I'll tell you on the test, it's going to be the pictures, the drawn pictures, so you won't have as much trouble. But for the lab, you have to know how to look at a picture and identify the structures, because you're going to have a lab practical, which means you're going to walk in here. I'm going to have a bunch of microscopes assigned. You're going to have a multiple choice question. You're going to have to choose, what is this? And you're going to have to identify what it is, A or B, A or B. All like 20 microscopes, 20 different structures. So you have to be able to identify these structures under a microscope, because you're going to have to do it live. So look here, and the neurons, you see those are individual cells. B is a blood smear. And there's 2 kinds of cells in B, aren't there? Do you 2 kinds of cells? What are those 2 kinds of cells that they talk about in B? White blood cells and red. What do we know about white blood cells? Anybody know what they are? Are there some characteristics that they talk about? The two are very important. Huh?
White
mobile, and what else? So, what? Oh, phagocytic cells, which means they're like amoeba, right? They're like amoeba. Remember the amoeba picture, the eukaryotic cell, the single-celled organism? They are, they're mobile, they're phagocytic with a pH. Say that again? That's right, they're big, they're bigger. In humans, they lack a nucleus when they're mature. Huh? Say that again? Red blood cell. I was trying to make it short. Actually, the when you see this word RBC on a test, it's red blood cell, right? It's not a word, it's an acronym, right? Humans lack nucleus in the mature. Red blood cell. Okay? Now look at, and of course the red blood cell, why did I put, goodness, I messed that up. This should be over here written, right? Sorry about that. So what about, which of these, let's take a look at these cells. When we're looking at that picture, there's two kinds of cells there, right? It's either a red blood cell or it's a white blood cell. When they say white blood cells are, let me say this. I'm going to say something, and let's see if we can understand the analogy. If we've heard before, and it's kind of true, males tend to be larger, faster, stronger than females of our species. Do you agree? We've all heard that. It's true. But does that mean all males are larger, stronger, and faster than all females? No. That means there's a range, right? There's a range. So when we say white blood cells are bigger, that really means what? They tend to be bigger, right? They usually are bigger. Does that mean that there couldn't be a red blood cell, or white blood cell is smaller than a red blood cell? Absolutely not. That's possible. But there's a range that they could overlap, but most of the time they're bigger. There's two kinds of cells there. Do you see two different kinds of cells? Let me sketch out the two different kinds of cells. What I see is kind of something that looks like this with all kinds of stuff inside it, right? And these things, what am I going to call these things? What would I call them now that I'm educated on what it is? Well, yeah, you could call it white blood cell. What do I call these things? These kind of dark gray areas and light gray areas and kind of organelles, right? I would call them organelles, right? Probably the biggest one is probably a what? Well, the biggest organelle is probably a nucleus, right? Because if you studied the pictures of the cells, and by the way, that's the biggest deal here, is to know which cell's which, right? And what they do. That's the point of this whole activity, right? So you have that kind of cell, and then you have another kind of cell, kind of looks like this, depending on where you're looking at it, because some of these look different, and the shapes are a little different. That's because these are cut, right? So you cut them, and depending on where you cut them, you're going to get different shapes. But I don't see any nuclei, do I? I just see kind of like a homogeneous color. It almost, in fact, I could almost imagine looking at the photocopy that it's red, right? Which one of these things do you think have a nucleus? This one, right? So if these don't have nuclei, what are they? Red blood cells. Does that make sense? So when I look at B, and B is pointing to something, even though it's small, it's smaller than some of these big ones without any nuclei, I got to say B is what? White or red? A white one, a white blood cell. And C is pointing to another one that again looks like number one, another white blood cell, but it's pointing to the cytoplasm. B is pointing to the membrane, the cell membrane. Everybody agree with that? Cell membrane, where B versus C is pointing to the cytoplasm. Are we okay with that? Is somebody confused about that? It's very quiet in here. I don't know what's going on. How do we feel? They could be bored, but if they're bored, I hope they know what they're doing because their test is next week. Everybody okay? So you should be able to figure this out. You should have figured it out already. First of all, in the picture on page 76, the pictures, not the drawing, A is the nucleus of a neuron, right? B is the plasma membrane or cell membrane of a white blood cell. And C is the cytoplasm of a white blood cell. Good. Because it looks like this kind of cell. I divided it into two types of cells. Those that have nuclei and those that don't. All right. The next page, page 77, we went into a lot of detail on it. If you need help, I'll be here after school for a little bit today. Not too much time, though. 78, cell components. This should have been easy, guys. You should have been able to look at these and go back to the drawings and identify which is which, honestly. Some don't, but there are clues. Read the words. But there's only one that might be hard. But I'll go ahead and give them all to you. You ready? So B says, I'm going to tell you, is a ribosome. B is a ribosome. If you read the definition of a ribosome, which I know a lot of you don't like to read, you know what I'm saying? We went through the organelles. If you read through the little definitions of the organelle, it says there's a large and small subunit. It's the only one that has that definition. So you should have been able to identify ribosome. Where the ribosome is located where? I'm going to go ahead and give you an idea where it's located. But, well, actually, ribosomes are on the rough endoplasmic reticulum. Rough endoplasmic reticulum. Again, you need to do the reading. You need to read. Ribosomes are in the rough endoplasmic reticulum. And they're also found in the cytoplasm, loose in the cytoplasm. That's it. They're not found in the nucleus. They're found in the cytoplasm itself or on the rough endoplasmic reticulum. Now, I taught you that already. It's not only did I expect you to read it, but a week and a day, actually exactly a week from today, a week ago today, I drew a cell up here. I drew a membrane, cell membrane. I drew a nucleus. I drew an endoplasmic reticulum. And I drew a rough and a smooth endoplasmic reticulum. And I told you the rough endoplasmic reticulum is due to ribosomes. So rough endoplasmic reticulum, is rough because it has studded with ribosomes. That's what the bumps are. So ribosomes, they're on the rough endoplasmic reticulum and in the cytoplasm. What do they do? If you read, I don't even feel like telling you what they do because you should be able to look it up, right? Answer? They make proteins. Good. Should be easy.
Instead of spelling out loud, can I just put abbreviation for certain numbers? Because some of them, like the ones that are long, I just put like abbreviation, like the first letter of each word.
You can do abbreviations, but I want at least a few letters so I can recognize what you mean. Like for rough endoplasmic reticulum, you can put RER. And the smooth endoplasmic reticulum, you can put SER. I'm okay with that. Those are common acronyms for those organelles. So mitochondria is the next one. C is mitochondria. Again, if you would have looked at the pictures and you would have read about it, you would have read in the drawings of the organelles, when you looked at the organelles of the animal cell or plant cell, you would have read mitochondria. An organelle bounded by a double membrane system. The number in a cell depends on how metabolically active it is. And look at the drawing, it looks like that. It looks like a maze. They actually look exactly alike, the previous drawing, the label drawing in this one. I hate the fact that I have to actually walk you through this, because I think you could have done this on your own. What do mitochondria do? They help metabolize it. They convert the energy in the cell. They make food energy into ATP. That's what they're doing.
What is that?
It's an ambulance?
No, ATP.
Oh, ATP is the energy currency of the cell. I thought you were asking what that noise was. Yeah. They convert food into energy that the cell can use called ATP. Again, you can look it up. I said it, but it's on the video as well. So we're gonna move on. I want you to. You know what? I'm not gonna tell you all that. You know why? Because guess what you can do. Look right here. There's a picture, guys. Drawing, label, definitions. You should have been studying that all along. Okay, Golgi bodies, D is Golgi body. All right, it says transfer vesicles from smooth endoplasmic reticulum, secretory vesicles budding off. If you look at Golgi body, the definition from the previous page, I'm looking at 75 and 76. If you look at Golgi body, it tells you what exactly they do. The Golgi apparatus, it could be called Golgi body or Golgi apparatus, a series of flattened discs, shaped sacs, stacked one on top of the other and connected with the ER. The Golgi stores, modifies, and packages proteins and tags proteins so that they go to their correct destination. And there's the shape. Look at the shape, and you'll see it looks exactly like this drawing. Where is it? In the cytoplasm. What does it do? It transfers proteins and lipids from the smooth ER into out of the cell. Or rather, out of the cell or to other places in the cell. The next one is the endoplasmic reticulum. Both what? Look at it and tell me, what are both of those? Both the rough and the smooth. What are why do we call the rough? Look at it. Because they have ribosomes. They're all studded with ribosomes. It's a really great drawing, isn't it? And it's labeled really well. And you see it that's called the rough endoplasmic reticulum. And the smooth is continuous, isn't it? It's just that you stop seeing ribosomes after a while. And that surrounds, I showed it to you on a three-dimensional model on screen, right? It's actually all around the nucleus. The next is what? You could tell me what the next one is. Chloroplast. It looks exactly like the other. And of course, what does it do? It converts sunlight into sugar. Converts sunlight into sugar. And we'll talk about how it does that and all the different components of it later. Where are they? In the cytoplasm. Most of these organelles are going to be in the cytoplasm. Very good. The next is a lysosome and a food vacuole. Now this is phagocytosis. So when you eat something, how does that food get into your body? When you swallow it, it's not in your body yet. You have to get it in your body. Your cells have to absorb the food. After your acids, after your teeth, your saliva, the acids of your stomach and the enzymes in your intestines, break it up into tiny, tiny little chunks. Your cells have to do phagocytosis. And there is the drawing of it. Really pretty drawing. I think it's very clear. And it's labeled. You see it labeled? Phagocytosis of a food particle. And so that food particle gets absorbed. It could be a little tiniest chunk of protein. It could be a tiniest chunk of carbohydrates. It could be a tiniest chunk of fat, whatever. So it gets absorbed, it gets surrounded by a membrane and gets put into the cell as a food vesicle, the food vacuole. Your cell creates a vacuole, it's full of food. How's your cell going to digest that? How's the cell going to digest it? Here's this food vacuole sitting here. It has food. Maybe, you ate some eggs this morning. It's not an egg, right? Because it's a protein or a fat or something. And it's surrounded by a membrane because look at what happens. That's phagocytosis. We'll look at it again later. But how do you get to digest that? There's food in there. You have to break it down. Why does it just start breaking down? Why can't you have the enzymes here? Let's say you have, I don't know how to draw, so you'll excuse me if I draw horribly. So you got the enzymes. Why don't you just want the enzymes that destroy fats and proteins? Why don't you want them in the cytoplasm?
Because that's made out of.
That's right, because your cytoplasm is made out of proteins and fats. And if you have those enzymes running loose in the cell, you'll destroy the cell. So you need them in a vesicle. Now, by the way, when you die, the reason you start to rot is those lysosomes break apart, the enzymes get loose, and they break you down. Even if worms never get to you, if we opened up your coffin 10, 15 years, 3, 4, 5, 10 years later, there'd be nothing left but a bunch of mush if we get to your soft part and your bones, right? You wouldn't see organs or eyes, maybe some hair. You know, hair takes longer to break down, and your fingernails. But most of your soft tissues would be mush, if we get to that at all. So these enzymes have to be placed inside a protective barrier called a membrane, right? So we have to put it inside a membrane. So this is an organelle. What is this organelle called? This is the food vacuole. We have the food inside a membrane because we don't just want food everywhere just being disgusting. It has to be able to fuse like two bubbles. Have you ever played with bubbles? You ever seen two bubbles come together and make into one? Right? So that's called fusion. The membrane, bubbles are made of membranes, and they'll actually just fuse together. You've seen that happen, right? And in fact, we will be playing with bubbles, I think, next week. So there's a membrane, and I will not have a mess in the room. I don't want people slipping on people. Silly soap. Yeah. Exactly. This is the whole organelle is called the lysosome. So the lysosome has the enzymes that destroy, the killers. All right. The food is in here. This is called the food vacuole, or food vacuole. The lysosomes are just waiting for food vacuole. As soon as they come into contact, they fuse like two bubbles, and the lysosomes can start digesting the food. All right. So that's a lysosome, and what's their function? To digest food. And where are they in the cytoplasm? Yes. So next one is obvious. The nucleus, where in the center of the cell, in the cytoplasm, however you want to say it. It's not necessarily the center. We say center, but can it be off to the side? Sure. You know, where's your heart? About a little bit to the right of the center, right? Do you know some people, their heart's on the left side? On the opposite side? They're on the right side? Instead over here, it's over here. On their right, your left. It's weird. So, you know, usually our liver's over here on the right side. With some people, all the organs are reversed. Isn't that scary? I mean, there could be some issues, especially if you're having surgery. And the surgeon's like, what the? So, yeah, it could be. I can imagine the first time somebody saw that, you know. They cut someone open, they're ready to do heart surgery. There's no heart. What happened? You know, it's like, what's going on? But yeah, there are people that are born that are their organs placements reversed. So when we say the heart's on the left side of center, that's most of us, right? Your left side, right? But some people it's on the opposite side. It's really weird. Life is strange, guys. Life is strange. All right, so the nucleus is generally in the center, and there's a nucleolus and all that. Locations in the center of the cell and the cytoplasm. The function is to hold the DNA. And it has a nucleolus whose function is to hold the RNA. It actually replicates all the DNA is in the cell, is in the nucleus, never leaves the nucleus. And the function of the DNA is to make mRNA, but we'll talk about that later.
So it functions to hold the DNA and RNA?
It holds some RNA, but really the function of the nucleus is to hold the DNA. To hold the information, let me put it this way, to hold the information for the cell. The information for the cell. All right, so microtubules, you know what they are, obviously, right? They're microtubules, they're tubes, they're small, hence micro, right? So microtubules, look at them. What are they? You can tell me this. Don't tell me you don't know. This should be easy. Please look at the picture and tell me what they are. No, look at the picture of the cells, man, and tell me what those two little groups of tubes are. Somebody said it. Centrioles. That's right. Who said that? Oh, good. Centrioles. That's right. Centrioles. Look at them. There's two of them. They're tubes. On page 76, on the right side, the second word down is pointing to two things that look exactly like that. Centrioles. Structures associated with nuclear division. They are composed of microtubules. But if you see how easy this is if you would have used this to look at the chart, guys. And people are complaining to me that it's hard. I'm like, are you serious? Yeah, because you're not looking hard enough. I told you all the answers are there. I wouldn't give you something where I didn't give you the answers. I'm not going to have you like, you just have to find it and put it together. What's she doing?
I'm looking.
You're making faces or something. So I'm not going to tell you. You heard me. You can read the function, right? You can read that. What's the last one here? What do you think that is? The last one on this page. Let's see. If I read, it says basal body anchors the flagellum. So what is this thing? Flagellum. It says it in the problem. There, look at it. Basal body anchors the flagellum. It's the flagellum.
Where is that in the picture?
In the very picture. In the picture itself. Read it. It's the last part. You have to read.
I'm not talking about that picture.
Oh, where is the flagellum? It's not going to be in the animal cell or plant cell. Where would you find a flagellum? Where would you see it? Over in the unicellular eukaryotes, look at page 73. You'll see two sets of flagella. Flagellum is the one's plural, one's singular. We got it?
Yeah.
All right. So, and what is it for? Come on, what is it for? What's the flagellum for? To help move, locomotion. And where is it? It's along the outside of the cell. It's an extension of the membrane, if you will. All right, cell component. What are we looking at? The next one on page 80. The first one on page 80. What is it? Cytoskeleton. I mean, it looks like a skeleton, for Pete's sakes. I look at it and I think Halloween. All right. Somebody might say spider web, but there's no such organelle as web. Cytoskeleton. Looks like a skeleton. Look at it. You see it connects all the microtubules, microfilaments, and intermediate filaments all together make the cytoskeleton, holding the organelles in place. They act as highways. They move things around. So that is a cytoskeleton. Where is it? In the cytoplasm, right? It's in the cell. It's the fluid of the cytoplasm moves around it. What's the function? That's right. Shapes the structure, right? Gives it shapes. Allows your cells to hold their shapes. Otherwise, they'd be little blobs, right?
Same function as the next one.
Cell wall is a little bit different. It does help with shape and structure. You're right. But cell walls are outside the cell, first of all, and they help protect the cell, right? Remember we talked about plants and fungi and bacteria. They need help with water, regulating water, and they don't want to explode, so the cell wall holds them together, right? So they're really protective, structurally protective. Cell junctions is difficult. That's one you would have had to look up. That's why I have a Chromebook carton here. Although I'm giving you the answers, so I guess you don't have to look them up. Cell junctions are located on the edges of the cell. What do you think they do? Look at the picture and tell me, what do you think they do? They hold cells together. Do you ever wonder how your cells hold together? Look at your skin. You're made of trillions of cells. Why are they all sticking to each other? The cell junctions. They hold them together. They're held together by junctions, like tight junctions, by desosomes.
You said they on the side of the cell?
They're all over the cells. They're in between the two cells. So when you're looking at it, if you have a cell like this, You have another cell next to it, like this is your skin, this might be the cells inside your intestine. This has a nucleus, this has a nucleus. There's all kinds of organelles, right? The endoplasmic reticulum, the rough and the smooth, endoplasmic reticulum, rough and smooth, right? So all kinds of cells. Listen, you have these things called tight junctions that hold them together. You have, I'm sorry, am I interrupting you? So tight junctions hold them together. You have a desmosome that kind of sticks. They have kind of like, I want to say, little threads that come into and bond with the cytoskeleton on each side. Again, what's that doing? It's making sure you don't fall apart, guys. It's making sure you don't fall apart. Without these things, you'd fall apart. You wouldn't be. The cells wouldn't hold together. And then you got these things called gap junctions. What do you think they do? No. They allow for things that be transported from one cell to the next. Oh, you're not. Oh, you're done. Oh, too bad. We ran out of time. I wanted to go over centrifugation. Remember, this is all done. That should help you with page 82. And I will send you a video on centrifugation.
How come we never played with buckets?
Why do you guys?